Lipids. Lipids are the one class of large biological molecules that do not form polymers. So it means that a lipid molecule, it is not composed of monomers, right? So the molecules of lipid, it consists of carbon, hydrogen, and also oxygen. There are three groups of lipids. The first one is fats. Okay, so fat that is in solid form at room temperature. Usually it comes from animal fat or saturated fats. And then the fat that is usually in liquid form, it is from plants, okay, or unsaturated fat. It is based on the composition of saturated and also unsaturated fatty acids of the fat. And then second is phospholipids. So phospholipids, as you know, it is the major component of plasma membranes or membranes of organelles. And then the third one is steroids. So steroid, the structure of steroid is a little bit different from the structure of fat and phospholipids. Where the steroid consists of four fused rings. For fat and also phospholipid, it consists of two major components. For fat, it has glycerol attached to three fatty acids, and as for phospholipid, it also has glycerol and two fatty acids, and also another one is choline group. And then there's another group of lipids that we are not going to look in your chapter, which is waxes. Okay, so only these three groups of lipids that we are going to go through. The unifying feature of lipids is that all lipids have little or no affinity for water. So it means that lipid is not soluble in water, as it does not form hydrogen bonds with water. It is not lipids is not attracted to water molecule through the formation of hydrogen bonds. And then so therefore lipids are hydrophobic because they consist mostly of hydrocarbons which form non-polar covalent bonds. Okay, it will not form hydrogen bonds with water molecule. And then lipid dissolve in organic or non-polar solvents such as acetone, ether, chloroform, and also alcohol, but does not dissolve in water. Okay, so the first group of lipids is fats or triglycerides. So fat and oils are esters which form the condensation of one molecule of glycerol to three molecules of fatty acid by an ester linkage. So the glycerol molecule is connected to three fatty acids molecule through ester linkage, and the process of producing or synthesizing triglyceride is known as esterification. Okay, so which involve an alcohol, which is glycerol, and also three fatty acids. Okay, one glycerol molecule to three fatty acids to form triglycerides or triacylglycerol. So glycerol is basically a 3-carbon alcohol with a hydroxy group attached to each of the carbon atom of the glycerol molecule. And then the fatty acids consist of carboxy group attached to the long carbon skeleton. Okay, in this slide we are going to see the reaction that involved in the synthesis of fat. To synthesize a fat molecule, it is through the process of dehydration reaction or esterification. Okay, so in this diagram, as you can see, there is one molecule of glycerol. A glycerol molecule will have 3 carbon, 1, 2, and 3. Each of the carbon is attached to a hydroxyl group. And over here you have the fatty acid, in this case is palmitic acid, and as you can see here, all the carbons is attached to hydrogen atoms. So this is actually a type of saturated fatty acid. Okay, so because it is fatty acid, over here you have a carboxyl group. Okay, so in the formation of a covalent bond between glycerol and fatty acid, which is in the formation of ester bond between the glycerol and fatty acid, a water molecule has to be removed. So in order to remove the water molecule, hydrogen from the glycerol hydroxyl from the fatty acid will be removed to form one water molecule for every fatty acid that is attached to the glycerol molecule. So over here you can see one water molecule is removed for each fatty acid joined to the glycerol. So in order to synthesize one fat molecule, okay, one molecule of triglyceride, 3 molecules of water will be removed, as you will be adding 3 fatty acids. Okay, 3 fatty acids is added or attached to the glycerol, and so therefore you will remove 3 water molecule to synthesize one fat molecule. Okay, so as you can see, this is the structure of a fat molecule or triglyceride or triacylglycerol. Okay, so the over here you have the glycerol molecule that is attached to 3 fatty acids. Okay, 3 fatty acids. So the linkage between the glycerol and also the fatty acid, we call it as ester linkage. So you should be able to label the ester linkage between the glycerol and also the fatty acid molecule. Okay, so you have to know where to label the ester linkage. Okay, so fat separates from water because water molecules form hydrogen bonds with each other and exclude fats. Okay, so fat, as you know, it is hydrophobic. It is not attracted to water molecule. So when fat is in the water or is in the aqueous solution, it will not dissolve in water as it cannot form hydrogen bonds with water molecule. So fatty acids, which is the component of fat molecule, it varies in length based on the number of carbon atoms in that fatty acid molecule. Okay, and also in the number and locations of the double bonds between the carbon atoms. Okay, so uh, so the fatty acids, okay, fatty acid is the component of fat. Okay, one fat molecule consists of 3 fatty acids. So the first type of fatty acid here, we have the saturated fatty acids. So remember, fatty acid is the component of a fat molecule or triglyceride. Okay, so saturated fatty acids have the maximum number of hydrogen atoms possibly attached to the carbon atoms, and there's no presence of double bonds between the carbon atoms. Okay, so there's no double bonds and all the carbon is attached to 2 hydrogen atoms, as you can see from the previous slide. So such as, for example, palmitic acid, all the carbon is attached to hydrogen atoms. Okay, and there's no double bond. And stearic acids, so therefore, because there's no double bond, there's no formation of kink in the fatty acid molecule, so the fatty acid is a straight molecule. Okay, is a straight molecule. Alright, so saturated fats. So saturated fats is made up of one glycerol, okay, one glycerol attached to 3 fatty acids. Okay, 3 fatty acids. Okay, because we are looking at saturated fats, the saturated fatty acids which is the component of the saturated fat has to be made up of saturated fatty acids. Okay, saturated fatty acids. So these are saturated fatty acids of where you can see all the carbons is attached to hydrogen atoms. Okay, all the carbon have the maximum, okay, maximum number of hydrogen atoms, and there's no presence of double bonds. Okay, double bonds between the carbon atoms. Okay, so remember, fatty acids is the component of fat. Okay, so saturated fats are solid at room temperature because the molecule of fatty acids is straight. Okay, straight. So at room temperature, fat molecule will exist in solid form because the fat molecule can be packed close together to form solids, okay, at room temperature. So example of saturated fats are fats from animals such as butter, okay, butter or lard. So those are example of fats obtained from animal that is saturated fat and it is solid at room temperature. The next type of fatty acid is unsaturated fatty acids. So remember, fatty acid is the component of fat. Fat consists of glycerol with 3 fatty acids. Fatty acids, it is divided into 2, which is saturated fatty acid and also unsaturated fatty acids. Okay, so this time we are going to look at, for this slide is for unsaturated fatty acids. Unsaturated fatty acid have one or more double bonds between the carbon atoms. For example, oleic acid and also linoleic acids. Due to the presence of double bond between the carbon molecule, okay, so between the carbon molecule there is presence of double bond. So whenever a double bond is exists in the chain of fatty acid, it causes the molecule of fatty acid to be bent, or it forms kink. Okay, kink. The molecule of fatty acid will have a bent structure. Okay, so bent molecule due to one or more double bonds within the structure of the fatty acid. So the fatty acid molecule, due to the presence of the double bond between the carbon atom, so the carbon atom can still accept one more hydrogen atom. Okay, so the carbon atom in fat, it can either attach to the carbon atom like this. So this is in saturated fatty acid, or in the unsaturated fatty acid, it is like this. Okay, carbon, and also hydrogen, one hydrogen for each of the carbon that has the double bond. Okay, so uh, unsaturated fats. Okay, so unsaturated fats are fats where the fatty acid will have at least one double bond. Okay, at least one double bond. So whenever a double bond exists, it causes the fatty acid molecule to be bent. Okay, to be bent or have a kink structure. Okay, so fats made up of unsaturated fatty acids are called as unsaturated fats. So unsaturated fats are liquid at room temperature. For example, plant fats or oil from plants, and also fish fats. So unsaturated fats are liquid because at room temperature, due to the presence of the band structure here, it prevents the fat molecule to pack close together. Okay, to pack close together. So when the molecule cannot pack close together, so the molecule will not turn into solid and will be remain as liquid form at room temperature. Okay, so in this diagram, it shows you the difference between saturated fats and also unsaturated fats. So example of saturated fat here, you have butter. Okay, so butter, the structural formula of saturated fat molecule. So all the fatty acids, okay, of the saturated fat molecule, they are made up of saturated fatty acid. Okay, actually 3 saturated fatty acid connected to one glycerol molecule through the formation of, through the linkage of ester bond. Okay, and then for unsaturated fatty acids such as this olive oil, okay, so the structural formula of an unsaturated fat molecule, it has to have at least Okay, at least one double bond between the carbon atoms within the fatty acid, causing the fatty acid molecule to be bent and preventing the fat molecule to pack close together, and causing the fat to be in liquid form at room temperature. For saturated fat, because the structure is the structure of the fatty acid is linear, so it can cause the fatty acid to be packed close together, causing the fat to be solid at room temperature. Okay, solid at room temperature for saturated fat. Saturated fat is liquid at room temperature due to the presence of kink or band structure. Okay. A diet rich in saturated fats may contribute to cardiovascular disease through plaque deposits. Okay, so for example, if you eat too much burgers, so the fats from the burgers will lead, if you eat too much, it will lead to the formation of plaque in your blood vessels and leading to cardiovascular disease. Okay, the next one, hydrogenation is the process of converting unsaturated fatty acids to saturated fatty acids by adding hydrogen. For example, vegetable oil. Okay, so you know that vegetable oil is liquid at room temperature. Okay, this is because in the fatty acid component of the fat, there's at least one double bond between the carbon. Okay, one double bond between the carbon. So each of the carbon will have one hydrogen atom attached to the carbon that has double bond. Alright. So this vegetable oil will be converted. It can be converted into solid form through the process of hydrogenation, where the double bond between the carbon will be broken so that the carbon can receive another hydrogen atoms. Okay, so initially, due to the presence of double bond between the carbon atoms, so the molecule of fatty acid is bent. So therefore, when there's no double bond between the carbon atoms anymore, so the molecule of fatty acid will be straight. Okay, will be straight. So due to the structure of the fatty acid that is straight, so therefore now the vegetable oil is turned into solid form. Okay. So hydrogenation of vegetable oils also creates unsaturated fat with trans double bond. Okay, trans double bond. So this trans fats may contribute more than saturated fats to cardiovascular disease due to their configuration that does not produce bands at the site of double bond. Okay, for example, this is the structure of unsaturated fatty acids. Okay, so between the carbon there's a double bond, and then the presence of the double bond causing the fatty acid molecule to be bent. Okay, so this is known as cis double bond, which cause the fatty acid molecule to be bent or have a kink structure. Okay, so through the process of hydrogenation, okay, so this cis double bond can be converted into trans double bond. Okay, trans double bond. So this trans double bond, the carbon atoms, the two carbon atoms, there's still presence of double bond, but this time instead of the molecule, the fatty acid molecule to be bent, the molecule of this trans fatty acid will be straight. Okay, will be straight. Okay, the trans fatty acid, the molecule is straight. So by having a straight molecule, okay, the trans fatty acid have a straight structure. So this cause the trans fatty acid of this fat to be solid. Okay, at room temperature, and they can pack close together, leading to the formation of plaque deposit in blood vessels, which can also contribute to cardiovascular disease. Okay, So as you can see here, it is more solid. Trans fatty acids are more solid at room temperature than cis fatty acids. Double bonds still exist, but for trans fatty acid, it does not have a band structure. They have a straight structure. The next group of lipids are the phospholipids. So in a phospholipid molecule, it is composed of two fatty acids molecule, a phosphate group which are attached to a glycerol molecule. Okay, so the two fatty acid tails are hydrophobic. Okay, so the two fatty acid will form the hydrophobic tail. Okay, the hydrophobic region of the phospholipid, and then the phosphate group and its other component will form the hydrophilic heads. Okay. Okay, so this is the structure of a phospholipid molecule. So um, it has two major regions, which is the hydrophobic region, okay, the hydrophobic tail, and also the hydrophilic heads. The hydrophobic tail is made up of two fatty acids. Each of these two fatty acids is attached to glycerol molecule. So glycerol molecule, as you know, it has three carbons, 1, 2, and 3. So the first two carbon is attached to a fatty acids, okay, and then the third one is attached to a phosphate group, okay, a phosphate group, and the phosphate group is attached to a choline. Okay, so this, as you can see, the hydrophilic heads, due to the presence of phosphate and also choline, which have charges, okay, so this causes the hydrophilic heads to be polar. Okay, so because it is polar, it can attract water molecules, so therefore it is hydrophilic. Okay, they have charges. Okay. Okay, so when phospholipids are added to water, they will self-assemble into a bilayer. Okay, a bilayer with the hydrophobic tail pointing towards the interior. Okay, for example, here you have the hydrophilic heads, okay, and then the hydrophobic tail, and then you have another molecule of phospholipid. Okay, so the hydrophilic the hydrophobic region of the phospholipids will be attracted to one another. Okay, so this will be separated from the aqueous solution, from the surrounding water. Okay, and the hydrophilic heads will face the aqueous solution as it is hydrophilic, it is attracted to water. Okay, the hydrophilic head is attracted to water. So this is the basic arrangement of phospholipid in the formation of membranes. So the structure of phospholipid results in a bilayer arrangement found in cell membranes or plasma membranes or membranes of organelles. Okay, so phospholipid, as you know, it is the major component of all cell membranes. Okay, so basically this is the arrangement of the phospholipid when you place the lipid in aqueous solution or in water. So this is the hydrophobic region. Okay, so the hydrophobic tail will be attracted to one another and will be excluded from the surrounding water. And as for the hydrophilic heads, it will face the aqueous solution as it is attracted to water. Okay, so the last group of lipids are the steroids. Steroids are lipids characterized by carbon skeleton consisting of 4 fused rings. So this is the basic structure of steroid. It is composed of 4 fused rings. So steroids are classified as lipid because it is insoluble in water and solubility in nonpolar solvent. So it is soluble in nonpolar solvent. So example of steroids are cholesterol. Okay, cholesterol is an important steroids, and it is one of the component of animal cell membrane. So you can find cholesterol embedded in the plasma membrane of animal cell. Cholesterol also function as the precursor molecule to synthesize other steroid hormones such as testosterone, progesterone, cortisol, and also cortisol. Okay, so these are examples of steroids hormone which is synthesized from cholesterol molecule. Okay, so in these slides, as you can see, there's three different types of steroids. You have here the cholesterol, okay, which is consists of 4 fused ring, 1, 2, 3, 4, and then you have the side chain of the cholesterol here attached to carbon to this ring here. Okay, and then from cholesterol it can synthesize other steroids, other sex hormones such as testosterone and also estrogens. Okay, so this is the end of the video explaining on lipids.